getting to John. Well, as I said earlier, before some of you were on, John Bates, who has been Curator of Birds for 26 years at the Field Museum. We're honored to have him tonight. The last time he spoke to us, believe it or not, was February 2017, where he spoke about bird mold. John grew up in Tucson. He's a lifelong birder. He and his students study the evolution and maintenance of biodiversity, focusing primarily on South America and Africa. This research includes analysis of genetic and traditional museum specimen data to address questions at the tips of the tree of life for birds and other vertebrates. He's actively interested in training new generations of biodiversity scientists in the regions everywhere he works. And as Sunny said to him earlier, he's an unbelievable resource for those of us in Chicago. So, John, thank you for accepting the invitation. Somebody has on their phone. Is everybody muted? Or maybe that was John's phone.
I don't know. It didn't seem to be mine. I'm actually looking for my phone. I thought it could be. Let me see if I can get things up here and share my screen and get my talk up. So can everybody see that pretty well? So
Yes.
First, I want to thank Rena for inviting me, and Sunny for helping me with the AV. It's a real pleasure to be talking to you tonight. And as I set it up with Rena, I thought I'd talk a little bit about natural history museums and how we protect avian biodiversity through the science that we do. And so that's what I've put together tonight. Interesting. So some of you probably haven't been traveling around a whole lot over the last couple of years, given everything that's been going on. And here's the Field Museum in Chicago, and we're been just like everybody else, muddling through everything. When I talk about what we do in birds, I like to break it down into these sections. I mean, we do basic field work all around the world. The place I was most recently was Antarctica in January, and that's shown in that picture at the top there. And then we also have these incredible collections. We're one of the best natural history museums in the world, and that's my wife, Shannon Hackett, the other Curator of Birds at the Field Museum, and my partner in our efforts there. And then training is a big aspect of what we do, and you'll see that throughout the talk. This is a picture of me in Malawi a number of years ago, but we train in all kinds of different ways, from graduate to undergraduate to bringing in volunteers that are high school students, at least when we could before COVID. And then, of course, databases are a big part of what we do too. And I've spent a lot of the last year, even though we couldn't go into the museum a lot of times, on projects related to improving our databases of collections. And one of the things that happened during the course of COVID was that our databases got used more than ever, because people were still trying to get information, and they could do it because we had information up online. And the three things I have there on this slide, iDig, Bio, VertNet, and GBIF, are examples of what are called data portals. And if this is a whole area of active evolution in the way we share data with people. But these data portals share different types of collections data, often but we're all connected across the country in terms of museum collections as a result of these collection databases. Then finally, we have public programs and exhibitions, and our exhibitions are still there. The museum is back open to the public, and we continue to do public programs like this talk and others. And so keep your eyes out for various programs that are done by the Field Museum over time, because we like doing it, and we think we do a pretty good job of it. And then finally, there's research, and that's what I'm going to be focused on today. And our research has evolved tremendously just over the 25 years that I've been at the Field Museum. I mean, you'll see over the course of this talk that we can do different things than we ever could with respect to almost every area of research that we look at, whether it's genetic research to morphological research. And that's been one of the most amazing things to me over the course of my career, is watching how all this has evolved. So these are some of the folks, almost all the folks in our lab. They come from all over the world. We've got the guy looking into the freezer there is Brazilian. He got his PhD at the University of Illinois, Chicago, and is now a postdoc in the museum. Abhimanyu Lele up in the upper right-hand corner is an American citizen, but his family is from India. And so he spent a lot of time going back and forth to India over the course of his childhood. Taylor Haynes with the Pyrrhura parrot on his shoulder is a new student who's working on the genomics of parrots in lots of different ways. And the guy to the right of him is a postdoc named Chad Eliason. And he's one of the world's experts on understanding color from the perspective of how it's produced in feathers to how it ends up manifesting itself across plumages of birds. The woman on the left-hand side there down in the lower corner is Valentina Gomez. She's Colombian. She finished during the pandemic and has gone on to a PhD or to a postdoctoral program at Penn State University. And she works on the evolution of migration in a group of flycatchers and kingbirds, particularly fork-tailed flycatcher down in South America. So these are just some of the people that we have in our lab. And it's one of the great things about doing the job I do is getting to interact with all these young people and getting them introduced to all the new ways in which we can do the kinds of research that we do. So when I talk about the Field Museum, I like to think that while conservation is something that we do, we're basically focused on studying things like ecology and evolution. We're a scientific organization. But the truth of the matter is that in order to do good conservation, you need to have an understanding of the evolutionary underpinnings, and the same thing's true about ecology. And so all these fields actually overlap with one another and benefit extensively from what each field has to offer. And of course, in the museum, we have this additional facet where we can go beyond science and into art. And this is just one of Peggy McNamara's wonderful pieces based on what she sees in the collection on a regular basis as our artist in residence. And it's just a really nice picture of what collections can look like from the eye of an artist. And of course, with the exhibits, we continue to maintain them and improve them. We actually updated the what's now called the Ronald and Christina Gidwitz Hall of Birds, probably 6 or 7 years ago now, where we did a work redoing the entire panel system and bringing in a whole bunch of new information and updated information on phylogenetics and also natural history and ecology. So we're always trying to improve the products that we produce, and we're doing it in a lot of different ways. Now, again, like you all, I've been on Zoom for the last 2 years, and I happened to remember this from a 2000 talk I gave as museums were talking about what the new normal was. And, like everybody else, we've dealt with all these various aspects that are hopefully now coming around and changing a little bit. And so it's, the new norm is probably not going to be the same as the old norm, but we're getting back into the museum and working on a regular basis. The students are getting back in, visitors are getting back in. So things are definitely getting more like they were before the pandemic as time goes on. But we were talking about this before everybody else came on, and you have to admit that one of the things we've learned during this period of Zoom is that we can reach out to people in some ways that we never had the opportunity before to reach out to, or at least didn't think about. So this is a talk that Valentina Gomez gave to the Colombian Ornithological Society about 6 or 7 months ago on her work on fork-tailed flycatchers. And again, she's got upwards of 30 people there in a group that she probably wouldn't have thought about reaching out to before the pandemic. So there have been some benefits associated with how we do things. At the same time, it's pointed out a lot of other things. This is a woman named Lenika Strozier, and one aspect about our science was beginning to come forward before the pandemic was how much we had to do with respect to diversity and inclusion. And Lanika was a scientist that had worked in the museum for 8 or 9 years. And in March 2020, she died of COVID very early on in the pandemic.
Oh.
She had gotten 2 master's degrees. She was just starting as an instructor at Malcolm X College. And so, you know, this is driven home to the institution and all her friends, just sort of how all of this is affecting people in lots of different ways. And we need to do a better job with respect to building diversity and inclusion. And we're doing that at some level in memory of Lanika. And you know, these are the kinds of things that I know everybody dealt with across the pandemic, and it's important not to forget that. So over the course of the last 2 years, I've been spending a lot of time in a role that I was president of something called the Natural Science Collections Alliance, which is a consortium of museums that of people that work for the good of museums, primarily through actions in Washington, D.C. associated with talking to Congress people and writing letters to the National Science Foundation about various things. And one of the other things we've been doing is thinking about the next steps with respect to trying to promote collections so that they'll be continued to be supported in various ways. And one of the most recent concepts about this is what we call the extended specimen. And this started actually from an edited ornithology volume, which is on the left, which was done by a whole bunch of ornithologists, where they put together chapters about the various new ways in which bird specimens were being used. And you'll see this over the course of the rest of the talk, that this has changed dramatically over time. And this has taken hold and expanded out to basically all of biodiversity, where we're emphasizing that the way we collect specimens these days and put them into collections really makes them very different than historical specimens. And that doesn't mean historical specimens aren't valuable. It just means that there's new information coming in all the time. So when I look at the extended specimen, I look at all the things we have the potential to get out of a given specimen that comes into the museum. And these can range from the various ways we could prepare the bird to getting various types of tissue. We'll now have georeference data on where the specimen came from. We can do things like photos, field notes, molecular data of all kinds, stomach contents. There's post-collection stuff like CT scans, which I'll talk about. And then I'm also going to talk about parasites and potential pathogens. And these all result in science that we do, which is the other aspect of what I'll be talking about over the course of the talk. Now, one of the things that we always try to emphasize too is why specimens? I mean, we can do so much these days without specimens. And that's true, but you start losing data. And what I want to emphasize here is that all of this is complementary. So if you're only netting birds, you're not going to get that physical specimen that goes into the museum that allows you to handle it over time, but you can get an awful lot of information from a netted bird. And certainly we archive data today collected on birds that were not made into specimens, because a lot of times that's something that we either think is the best thing to do, or it's something that's required for a given region. And we still want to get research done and archive material from these birds. And then of course, we've got the rise of things like eBird, which are incredible assets to understanding lots of things about birds. But they're limited in terms of some of the scientific uses that they can be made to. And again, I want to emphasize here that what I'm trying to illustrate is that these are complementary things. You can get much more data in terms of photos and records via eBird, and people can participate in lots of different ways. And the key thing in all of this, though, is that there's data that you can't get from these kinds of approaches. And there's a complementarity that I hope will show up across the rest of the presentation today, too. So again, extended specimens matter. There are lots of questions like change over time, all these listed here. And the ones in orange are ones that I'm going to touch on today through some of the research that we've been doing. And the other thing I say there is why series matter. And this is again about having enough individuals to ask specific kinds of questions. And that becomes really important with a lot of the science we do. And anyone who comes into the museum will often hear me say that when you look at our series, it's a little like trying to do a medical study on some drug, where you wouldn't want to try that drug out on 2 or 3 people before you turned it loose on the public. Just in the same way, you need to have multiple individuals of specimens in order to begin to understand lots of kinds of questions that we're interested in. So here's an example of a series. And again, I apologize that this may be a little bit off-putting, but these are salvaged birds that have come from Wisconsin. And Dave Willard has worked up with Bay Beach State Park for a number of years. They have a rehab center, and they'll bring in birds like this. And what I want to point out here is these are all robins, and they're all different stages of robins. And this is the kind of information that literally doesn't exist for probably 98% of the world's birds. And yet you can have this kind of information about a common species like robin. And that allows you to do an awful lot of neat science associated with development in ways that are still beginning to evolve. So here's an example of one of the ways in which we can look at morphology that didn't exist 10 or 15 years ago. We have an NSF grant called OVERT, and we're CT scanning bird and amphibian and reptile specimens from our collection, and then making those openly available to researchers through another data portal that's called MorphoSource. And what you can see on this slide is that we've got a skull of an emu. To the right there's a Jasana, full-bodied Jasana specimen, head of a Chilean flamingo. And then one of the last ones here down in the lower right-hand corner is a loggerhead shrike that's 14 days old. And this is part of a series that was collected back in the 1960s as someone's master's student, and has been sitting in the Field Museum's collection for all that time. And we're now able to go back through and CT scan it and make that collection broadly available to people via this format of MorphoSource. So these kinds of approaches are revolutionizing our ability to look at morphology and look at evolution in terms of change over time when we have the specimens in order to do that. I wanted to mention peregrine falcons really quickly because as Rena pointed out as we said in the blurb about the talk, this is one of the peregrine falcons represent part of one of the best examples of how bird specimens have been used for conservation. And of course, I'm guessing you guys have heard Mary Hennon talk over the years about her peregrine falcon program, where she's monitored the local peregrines in the Chicago region for the last 35 years or so. That picture with the brooms was taken actually last week at St. Mary's Hospital, because Mary wanted to move the nest box, and she said the female was especially aggressive there. And so I'm the one holding the blue broom. And I can tell you that I have rarely been as concerned about being out around birds in my life as watching this female peregrine dive bomb us repeatedly as we were trying to move this nest box around. But peregrines are an incredible success story in a lot of different ways. And this is the primary piece of science that actually started turning the whole process around. Back in the 60s, people began noticing that peregrine falcon numbers, bald eagle numbers, osprey numbers were all going down. And they began to look for what was going on, and they noticed that there was no nesting success because eggshells were getting thinner and the females were cracking the eggs. But what they wanted to show was they needed to prove it. And what Hickey and Anderson, two scientists from the University of Wisconsin, did was they went around to a whole bunch of museum collections around the country, and they gathered eggshell thickness on a bunch of different species, some of which they thought were affected by DDT, some of which they thought weren't. And they measured pre and post era thickness in these eggshells, and were able to show dramatically that birds like herring gulls, it says here in 1967, shell thickness in herring gulls from 5 states decreased with increases in chlorinated hydrocarbon residues. So they were able to link up the eggshell thinning with chlorinated hydrocarbon residues. And they, in this paper using peregrine falcon eggs, they went around to collections like ours. And so, for instance, these birds that were collected in these eggs in 1904 by Arnold in Montana were part of the group that represented the control for their assessment of eggshell thickness in peregrine falcons. By that, I mean that this was a region where DDT wasn't thought to have played a big role, and it didn't. But in other areas like the East Coast, they could clearly show that eggshell thinning was actually happening in peregrine falcons. And this led to the banning of DDT. And of course, what's happened since then is bald eagles, ospreys, peregrine falcons have all made dramatic comebacks. And peregrine falcon was the last one of those birds to be removed from the endangered species list. And this can be applied in lots of different ways as we get new techniques. So another thing we've been able to do is look at isotopes for all kinds of questions. And isotopes are just variations of elements, and you can use them for all kinds of different things. And this was a really interesting paper that was published in 2015, which they came to us and they said, can we get some feathers of your ivory gulls? And we specifically want ones that were collected in the 1880s. And they put together a data set across the country where they looked at specimens, and they were able to get a time series dating back from 1880 into the present, and shows that over a 130-year period, mercury levels have been going up in this bird that is breeding up in the high Arctic. And so this is the kind of data that has real implications for conservation, in the sense that it's clear that you've got documentation that mercury levels are going up. They can come up with no explanation outside of human activities to explain this. And yet at the same time, there's still a lot of work to do to fully understand what's going on and what the ramifications are for these birds. But if you've got things like mercury that can show up in the high Arctic, you can pretty much guess that it might be affecting other ecosystems too. And so since this publication, we've had requests for feather samples from our specimens of rusty blackbirds, which is another bird that's declining rapidly in the Midwest, and people are really concerned about. And one of the suggestions is that they may be dealing with issues related to mercury on their breeding grounds. So these are the kinds of things that once one of these publications comes out, other people can look at this and they can begin to build on this research volume. So I wanted to bring up a bunch of things that, we haven't been dormant over the course of the pandemic, and we've had a number of students defend their dissertations. This is from Heather Skeen's dissertation in February 2022, and you can see the way a dissertation starts out with is the student will present the chapters that they've looked at. And so Heather was looking at microbiomes and malaria in migrant birds, and she was able to use the birds that have come in through all the salvage that's been done by the museum and working with folks to look at microbiomes of Catharus thrushes and look at malaria in Catharus thrushes. And what we know then with microbiomes is that there's data from humans primarily, but it's beginning to spread out to beyond humans, that suggest that microbiomes can have major influences on the behavior of individuals and on the behaviors of populations. And so it can affect things at all kinds of levels, which is what she's showing here in this picture of this starling and various aspects of its natural history and biology. And so this is what her dissertation was about. And she was trying to look at it across the annual cycle. And what I want to show you now is a paper that we were part of, Shannon and I, that Heather did. And it was partnering up with some really great colleagues of ours, Nathan Cooper and Pete Mara. Pete was at the Smithsonian Migratory Bird Center at this point. And they'd been working on Kirtland's warblers, and we got to talking to them and we said, what about studying the microbiome of these birds? And they said, yeah, let's do it. And if you know Kirtland's warblers well, you'll know that they breed in northern Michigan, a little bit into Ontario, and then they winter down into the Bahamas. And Pete and Nathan were specifically working on the island of Cat Island in the Bahamas. And so Heather went to these places, and one of the things they were doing is using new technologies of putting geolocators on these birds. And the interesting thing is that Kirtland's warblers have a small enough population that they were able to put these geolocators on in the Bahamas, and that they work off of cell phone towers. And so you can set up portable cell phone towers in each area. So they would do this in the Bahamas. The birds would migrate back to Michigan. And in many cases, or a number of cases, they could actually locate the same individuals again in Michigan. And so what Heather was able to do is sample the microbiome of the same individual bird sometimes at 3 different points on the annual cycle. And so if you think about this, I mean, this was unimaginable 5 or 10 years ago. And it relies on a whole bunch of different technologies, the ability to characterize microbiomes using genomic techniques, and the technology of being able to put transmitters on birds that you can actually get the birds to carry around, migrate with without any problem, and then actually find the same signal in very different parts of the bird's range. And this is not something that you could do probably on almost any normal species. One of the reasons why it works on Kirtland's warblers is because the species has such a small distribution, both in the winter and during the breeding season. So what do these things show us? Well, they show us that there are some types of bacteria like cyanobacteria, which is yellow on the graph above, where you can see that it's fairly frequent in small amounts in the Bahamas where the initial captures were done, and then less common when the birds first get to Michigan, and then absent actually in the later samples in Michigan. And cyanobacteria, turns out, is bacteria that's primarily or often associated with saltwater situations. And we think they pick up the bacteria in the Bahamas on their wintering grounds, which are almost always near the coast because the islands are fairly small. And then they're clearly shedding it over the course of the breeding season. If you look down below at this figure, what it's trying to illustrate is where there are significant differences between seasons and some of these other groups. And where it says NS, it means not significant. And you can see that some of these other ones are significant too. And some of these, like Firmicutes, is a type of bacteria that tends to be related to breaking down cellulose, which makes perfect sense because one thing that's known about Kirtland's warblers is that they tend to eat more fruit in the non-breeding season. And so you begin to see some of these basic aspects of natural history coming out in the microbiome of these birds. And again, the one thing I'd emphasize about this from a collection standpoint is now that we have these data, we can begin to look at how this changes over time. So this is really exciting work, and it's just been really fun to watch Heather get to do all this and publish it as part of her dissertation. Now I want to transition to another use of specimens, which was something that Dave Willard and I had argued about for a long time. This is one of our drawers of red-headed woodpeckers. And if you'll notice, if you look at these individuals, you can see that the white bellies are not as white on some individuals as they are on others. And this was something that if you pull out drawers of lots of our common Eastern birds, you could see a pattern like this. And Dave and I used to talk about it, and Dave would always say, you know, I think it's probably just that some of these birds weren't stored very well in the previous collections they were in before they came to the Field Museum, and they got sooty. And I don't know, I'd always kind of wondered about that. And I was talking to my graduate student, Shane Dubé, one day, and I said, you know, if you're looking for projects, you might look into this. And he got some money from an interesting program at the University of Chicago that hooks up graduate students from the sciences with graduate students from the arts. And he hooked up with a history PhD student named Carl Foldner. And together they put together this project where they started photographing individual birds and measuring the sootiness of these birds relative to a control. And they ended up with an incredible finding about what these sooty specimens actually tell us, which is basically the history of coal production in the Midwest. And so if you look at this graph here, it goes from 1880 to 2000. And the dark line is, the black line is the soot levels on birds through time. And the purple line is, can't see everything here, is actually soot production. It's a measure of soot production over time that they were able to find. And then the bottom line is another measure, and I can't, you can see it, it's it's how much coal is burned in the US. And there are two aspects of this story that are really important, because you can see that orange line actually goes up between 1960 and 2000, but the soot on the birds doesn't. And if you look at across the top, you can see various things that happened across the North American society to mitigate things. So for instance, after the Great Depression, there was a big drop in coal usage and in the amount of soot on birds. Then World War II comes along and it goes way up. And then you start getting pollution control acts. And what you can see in this is that despite the fact that we were using more coal, we'd actually developed methods of controlling the production of coal dust such that this was not getting into the environment in the same way that it had back in the early 1900s. And so I love this as something that was in our collections all the time that we actually didn't realize until two people had the thought to spend enough time and do it in a organized way to get these data. But two, that it really is a wonderful example of how we can illustrate that once we make decisions that are actually intelligent with respect to keeping the air clean, for instance, you can see those differences in lots of different ways. And so this was a really spectacular study that I think has some really important lessons for conservation. So another thing we've been able to do in the field, and this is related to this extended specimen concept, is start getting samples that can be used in various studies of parasites and pathogens. And we were doing this 8 or 9 years before COVID. And I don't think you couldn't come up with a more dramatic example of why science matters than what's happened in this pandemic from my perspective. And these kinds of diseases are out there in lots of different ways, and biodiversity has been dealing with these diseases for a long time. So the more time we spend thinking about how biodiversity copes with various aspects of viruses and bacteria and how it's transmitted, the better off we're going to be. And so this is something that we think collections can be extremely important for over time. And other people do too. You know, this is a photo from an expedition 3 years ago in Malawi, where one of our colleagues, Sadiq Waswa, is handling a bat, and these guys are collecting samples for Zika and other pathogens. We just had a pair of researchers associated with the museum come back from fieldwork in Togo, where they were doing the same kind of sampling, and geographic sampling is going to be incredibly important in terms of trying to get ahead of what's happening with respect to the changing landscape of viruses and other disease-causing agents over time. And then, of course, there's kind of the bread and butter of what collections do. And one of the things we continue to do is put together data sets that allow us to understand where undescribed biodiversity is. And you can see that this is a paper that came out in 2021. The research had been done actually about 7 or 8 years before by Sidney Melo dos Santos, who was a Brazilian, was working on his dissertation, gathered all his genetic data in the Pritzker lab at the Field Museum. And then finally felt confident to do all the analytical work on the data that he had to put it together during the pandemic and get out this review of variation in the black-capped and tawny bellied screech owl complex and describe two new species. And the first one of them that I'm going to mention is called Xingu screech owl, which is Megascops stangeyi. And I wanted to mention this because one of the things that's happening as a result of the realization that there's a lot of colonialism in science is that names of birds have often been very circumscribed to a limited number of people, namely white men for the most part. And there's been a lot of pushback on this, justifiably. And as this was happening, this paper was coming out, and I was thinking, that's interesting because we named this paper, this bird, and this was Sydney's idea, for Dorothy Stang. And Dorothy Stang was a missionary from the US. She was from Ohio. And she spent her entire missionary career down in the southern edge of the Amazon basin in an area where she completely fell in love with the culture of the people. And she was arguing very forcefully with the landowners and ranchers in the area. And in 2005, the ranchers hired an assassin to kill her. And she became an incredible martyr for the conservation movement and the protection of the rights of poor people in this part of Brazil. And so I put it out there only because I completely agree that we might want to get rid of eponymous names, which are naming things after somebody. And yet at the same time, I think honoring somebody like Dorothy Stang, which was, again, Sydney's idea, is just a monumentally valuable thing for a lot of the work that people you might not know otherwise actually do.
Wow.
So here are the distributions of the two owls we described as species in this paper. So stangeyi is found in this little area between the Rio Xingu and the Rio Tapajós in Brazil, in that sea. And then the other species is called Alagoas screech owl, and it's found over on the northern edge of Atlantic forest. And the thing about this bird is that this is a bird that's highly endangered. The forests in this part of the world are extremely fragmented, and so recognizing these species is really helping to highlight the need for better conservation in this part of Amazonia. And these are the kinds of things that just become really important. And I've got a couple links up here. I didn't want to try to make the talk get bogged down in doing this, but if you want to hear what they sound like, they sound different. There are measurement differences. And so this is kind of the bread and butter work we do. At the same time, this bread and butter work includes describing species that I never thought I'd be involved in describing, such as platyhelminthine flatworms. This was from a trip we did to Cordillera Azul National Park, and Caitlin Patitucci of the University of North Dakota did this for her master's degree. And you'll notice that one of these things is called Uvula Batesii, which is, again, an eponymous name for an old white guy. But at the same time, the other one is named after a woman named Tatiana Pequeña, who's been the longtime director of Cordillera Azul National Park. And so again, I think there's some interesting aspects of eponymous names that can really honor people that we should be talking about, I think, over time. At the same time, I think we probably will stop using eponymous names for the most part in ornithology from here forward. But I also want to emphasize how little is known about these animals. So these have both been described from a single specimen of Chlorocereile indo, green and rufous kingfisher, that was collected in Cordillera Azul National Park. That's the only place they're currently known from. And so they're out there somewhere in biodiversity, living in the gut intestines, but we don't know how many species they live in. We don't know what their geographic range is. And so there's an awful lot of work we have to do on some aspects of biodiversity, like these platyhelminthine flatworms. Now, you guys all know probably Dave or know the Chicago region migration research, but maybe you don't know about some of the recent stuff that we've done. I mean, this is a data set that's been built up over 40 years, and it's because of that, these time series allow you to do different things. These are the collision monitors. This is a 2010 photo. It's a much bigger group now, but they're going around the buildings every day and taking the live birds out to the rehab centers, and we end up with the dead birds. And it's an incredible database on migration through the Chicago area. And this is where sample size becomes so incredible. So this is some work that my former graduate student, Ben Winger, and his colleague Brian Weeks at the University of Michigan had been working on with Dave for a while now. And these are some of the highest, the most commonly found birds in the migration sample. And what this figure shows is over time, what's going on with tarsus length, which is a measure of size in these birds. And you can see that for every single one of the top 9 species, body size is actually going down over this time. And this is a very, very interesting pattern because the data are so good and the sample sizes are so good for these birds. And so one of the big questions is what's happening with this? If you look at what happened, what's predicted by warming temperatures, you can make a cogent argument that body size would go down as temperatures go up. And seems to be happening in lots of different birds. So that may be an explanation for this. At the same time, because this is a scientific effort, Ben and his colleagues have been looking at other possible factors. And so they're not entirely convinced that climate change is necessarily causing this pattern, but it's definitely consistent with it. And they want to do more delving into it, looking at some additional variables to try to see if they can tease apart things once and for all so that we have good data for this. But again, having this longitudinal sample with really great sample sizes for common birds allows us to look at these patterns and develop really strong patterns of what's going on. So the last thing I wanted to mention is a paper that we just got accepted into the Journal of Animal Ecology. And this was a paper that I dreamt up a number of years ago because I was working on the egg collections. And it's about climate change affecting bird nesting phenology. And we're looking at contemporary field records and historical museum nesting records. And we used a number of different data sets. We went to the Western Foundation of Vertebrate Zoology, and we also got data from the Chicago Academy. We had a lot of help from people. But what we were really building on from the historical perspective are egg collectors. And just really quickly, egg collectors were amazing natural historians. So they would go out and find these eggs. If you look at these cedar waxwing eggs here, you can see that they were collected in 1897. Let's see. Yeah, my eyes are bad enough that I can't read the date, but anyway, there's detailed data on this in terms of where it was. And this is up in Glencoe, just so you know. So it's definitely in the region. And here's one that's a little more legible so you can look at this. So this is one of my favorite ones. This is a upland sandpiper that was collected at Crawford and Devon in 1934. So you can start realizing that things have changed a lot in this area. And it was collected on May 28th. And what we can do with that information is it says incubation was 2/3 done. And there were 4 eggs collected. And so we can actually backtrack with that calculation to estimate when the first egg date was. And so you can see here, there's 4 eggs. That probably took about 7 days to lay based on the natural history that we know about upland sandpiper. They were probably 2/3 into incubation, which means that that's probably about 18 days. And so we can estimate that May 3rd was the date the first egg was laid. And because there's fairly large sample sizes of historical egg collections. You can do that for lots of different species from the Midwestern area. And so this was the data set that we put together for this project. We had eggs from Cook, Lake Kane, McHenry, Kankakee, and Will counties. So this is a very Illinois-focused, Northeastern Illinois-focused data set. And the historical data set had 1,550 egg sets of 72 species in it. Now, what do you do for modern data? So this is, to me, in some senses, as good as the collections are. I know how good the collections are. But I also happen to know that I had some colleagues that I'd gotten to know in my time in Chicago who had done a lot of field work. And finding nests is not easy, and most people don't do it today. And these data are from these older historical collections are actually fairly unique for most species of North American birds. But I happen to know that Chris Whalen and Bill Strasburger had done a lot of work. Bill did his dissertation at UIC studying cowbird parasitism out at Morton Arboretum. And what he would do to study that was go out and find every nest he could every single year. And so he wasn't collecting them, but he was collecting detailed data because of the parasitism work on the same kinds of things that the early egg collectors were taking, how many eggs they were, when he found the nest. And so we could make this same calculation of first egg date. And that's what we did. And so this gets into the weeds here that I'm not going to spend a whole lot of time about, but we also wanted to match things up to climate change. And of course, when you think of climate change, you think of temperature. The problem with temperature is that it's very hard to get clear average curves for temperature. And they don't go back, there aren't temperature records going back before 1900. So we had this data set that went back to the 1880s. Well, what we could use, we realized, was actually information from ice cores, because those actually give CO2 data, which tracks pretty well with respect to temperature. And we did this big analysis just to try to look at what was happening and what birds were doing. So long story short, it's not entirely clear with respect to the patterns we expected. So for instance, we expected that resident species might not be as affected as long distance migrants. And you can see here that basically the percentages of resident species and short distance migrants and long distance migrants that are affected are not really all that different. But overall, there's significant advancements in egg laying by about 10 days over this 130-year period. And so that's substantial. It's clear that it's changing, but it's also highlighting that it's not a simple a pattern. And I think we need to expect these kinds of things in a lot of the ways and with a lot of the issues we're trying to look at. So for instance, you've probably all heard the information about analyses suggest that upwards of 3 billion birds are missing from the North American avifauna based on data from breeding bird survey data and other monitoring like that. Well, the next question is, are all those birds being affected by the same things? And the answers are almost certainly no. And so we need to do a lot more research and get data sets like this to begin to understand and tease apart why things are responding the way they are. And one of the most important things with respect to that I always emphasize is that information needs to be archived in collections, and museums needs to be archived, and collections are a really logical place to do it, because that's what we've been doing for upwards of 125 years now. All right, so I'm going to stop here. I'm welcome to take any questions in the chat. We continue to work locally, nationally, and internationally. Museums have gone through a lot, but they've gone through a lot in the past, and everybody else is going through a lot. I think we'll see museums into the future. And as Rena said, I did have this other talk that I might be able to give down the road, and this is a little bit of a teaser as to what it might be about.
Oh, so you're not letting me keep the secret.
Nah.
Okay. All right. Well, okay. Now that you've shown it, though, then you have to promise. We will be in touch on that. Okay. There are some messages in the chat. Thank you, John. Very interesting.
So
Yeah,
let's, I can
I
go.
was just gonna say, for those of you who have not read The Feather Thief, and the fact that all of these birds in this collection were lost by this, I think, 19-year-old crazy person. It's a really good read and a very sad read. I know I talked to Josh about it. He said everybody in the field bird collection, bird division had read it. Okay. So let's go to some, I put some of my questions in the chat so I wouldn't forget them, but let me start with Bob Stanley, as he had a question that I had not yet put down. He said, I was pleased to hear that you used and were able to get good data from specimens from the 1880s. Is there a limit to the useful lifespan of the museum specimens for research purposes?
That's a wonderful question. So the real limit on older specimens tend to be the actual data they have with them. One of my favorite birds in the collection is a greater bird of paradise, which we pull out and show people. And the tag says old specimen collected by Wallace, Aru Islands. And Wallace is Alfred Russel Wallace. And so that sounds really neat, except that somebody clearly wrote that on the tag. It wasn't Wallace who did it. And older specimens tend to have a little bit less data. The localities can often be vague. But with all the new techniques we have, we're routinely going back now and say, taking a toe pad of a bird and getting genomic data on that individual specimen and what its DNA looks like. And we can always go back and measure them. And we can, now we could, for instance, we could CT scan them. Chad Eliason, who I mentioned at the beginning, is a postdoc working on kingfishers. He was doing a project on where he was trying to get access to skull morphology for all species of kingfishers. Well, ideally that would mean using skeletons or alcoholic specimens, but there aren't alcoholic specimens of a lot of species. So for example, there's this really cool specimen or species from New Guinea called shovel-billed kingfisher. And in order to get a image of the skull, we took a specimen, a regular study skin, because we leave the skulls in the study skins. And this was a bird that was collected in the 1930s. And it was taken down to the University of Chicago, and we were able to CT scan the skin, and Chad was able to get the data that he needed for his data set about the skull from that CT scan. So there are actually a lot of ways in which we can use older specimens into the future. So Rena asks, how did the collections help distinguish the two owl species? So a couple different ways. So I mentioned that vocalizations were different, but morphology
but
is
they're
all.
dead.
What's
So
that?
you can't, they're dead. You can't get that from the
So
specimen.
so a lot of that is done by recording the birds at the site and then collecting them sometimes in those situations. And then with this collected specimen, what you're getting is you've got the morphology, and you can do the comparative work too. That's one of the other things about having specimens side by side, is you can compare these birds from these different populations. So if I go back to that, what you'd see is that there are these different populations of things between the major rivers in Amazonia. And Sydney didn't think the other ones were quite different enough in terms of the combination of morphology and also genetic data to call different species. But these two populations were absolutely the most different in terms of plumage and also in terms of vocalizations. And so the plumage and genetic data are all coming from the specimens. So why is the flatworm species named after me? So it's named after me because the guy we collaborate with is a guy named Vasil Tkach, who's a absolutely fabulous flatworm specialist at the University of North Dakota. And one of the things we do is, because he is who he is, we often find the money to pay for him to go on trips. And I think he thinks it's the best thing to honor various people that are paying for the trips when it comes down to it. So that's why that happened.
Okay. I'll let you, as long as you're doing it, go ahead, read through the chat.
Yeah,
questions.
sure.
We'll just take a few more. So yeah.
Who would argue that museums are the logical with you that the museums are that's so Sunny asks, who would argue with you that museums are the logical data repository? That's an interesting question. I mean, over the years, there have been a number of stories where people lose track of the fact that museums are the focal source of the original data and start using it for other things. And again, that's not necessarily a bad thing, but I think that in the particular case that I was trying to focus on with the egg laying date stuff, actually the ecological community is, to me, one of the worst offenders with respect to not archiving data. And I find that fascinating. And that is that they go out into the field and they put together these data sets, but they're working on contemporary questions. And so they don't think about the long-term value of their data for the most part. Now that's starting to change, but that's something that our community's been doing since the inception of museums. And so people are coming around to the recognition of doing that, but it's hard to do it. And I'll frankly say that the museum community sometimes is their own worst enemy too, because we will say that for instance, if there's not a specimen, there's nothing to archive. And what I've come around to is that what we want is for data to be findable over time. And if you've got data that are collected over 4 decades, and it's collected in different ways, but it still allows you to look at the same thing, it would be really good to have people not have to hunt and peck for this stuff. And so with Chris Whalen and Bill Strasburger's data, they've published a number of papers on that information, but they've really never done anything with the egg laying data. So that's actually a brand new use of the data that they gathered, but nobody would have been able to find that until I asked them to think about whether or not that would be useful, and it turned out it definitely was. So I think both sides of the equation are seeing the need of archiving this, and the key thing is to make it generally findable for people. And this is a big conversation piece in the museum community, because the other thing is that we've always been focused on the collections themselves. And now we've got this whole big world of data, and data can grow really fast, and somebody still needs to curate it. And finding the money and positions and expertise to curate those data is kind of the next big hurdle for museum collections. And I think it's a challenge that we're still trying to figure out how to face. So let's see, Nicole asks, what do you think about neonics, how to reduce? So neonics, how to reduce? Do you mean by resources? Is Nicole here? Can she? I don't know the term neonics. Is it possible? Sunny, can she?
The pesticide. She means that's not the word.
Oh, neon. Oh, so I mean, humans have an unbelievable capacity to come up with things that have the potential to do bad things to the environment. And I guess, many of these pesticides come out with initial seals of approval, and you only really find out based on longer term usage and what's going on. So, DDT, I've actually had one of the collection managers of insects point out to me that DDT is one of the most effective things against bed bugs. But the problem was DDT became used for literally everything. And so it was so dramatically overused that it caused all these problems. I don't know specifics about neonics. I can look into that. I'll give you another local example right now that I'm wondering about. Over the past 3 or 4 years, I've picked up at least 4 dead great horned owls in places like Memorial Park Cemetery and Perkins Woods and Cavalry Cemetery in Evanston. And people will tell you that rat poison is not poisonous beyond rats, but I don't know how much you might have seen this in the news. There was this barred owl that showed up in Central Park, And it was actually hit by a driver who was driving through the park in the early morning, who picked it up and brought it to some official. And they ended up testing this owl for, I think it was the 4 different common kinds of rat poison. And the bird had very high levels of 3 of the 4 types. And I mean, it was hit by a car, so maybe it was perfectly fine with those kinds of rat poisons in its body. But I think we have to ask ourselves, what do we really know about the accumulation of things like that in these kinds of organisms? Because we just don't handle that many of them. And so I'm looking forward at some point to trying to test some of the tissues of these great horned owls and see what they look like relative to this barred owl that was in Central Park.
So we have 2 more questions in the chat. So let's do
Yep.
those, and then and then we'll let John go.
Can you make assumptions on eggshell thickness with eggs where the shells are completely intact, or do you need actual broken eggshells? Really interesting question, Alan. So our eggs in the collection are blown. And what that means is these egg collectors were really good at drilling these tiny little holes. And if you induce air, you can actually blow out the entire contents of the eggs. And I actually was in communication with Anderson, one of the 2 authors of the original work about where his data were, which it turned out they were actually at a museum. They were out in the Western Foundation of Vertebrate Zoology. So I was able to get access to his data sheets. But one of the things he said to me was he thought I was actually calling him about damage to eggs that he might have done while they were trying to look at it. And what I would tell you is they were able to get eggshell thickness data from these tiny little holes in peregrine falcon eggs that are no more than about 2 or 3 millimeters across. So there are ways to do that without affecting the egg. At the same time, and one of the things I would say is the neat thing about eggs is you can get data from the pieces of eggshell too, if that's all you have. And so there's a lot of neat things you can do with eggs. So let's see, the last question is, does the content of intact eggs just desiccate over time? That's an interesting question too. Again, what the collectors try to do is actually get it out, but it can actually desiccate in there, and parts of it do. And my favorite example of recent DNA work that's been done on historical egg collections was a paper that a guy did where he went around and he got permission to get very small samples of all the Labrador duck eggs in North America. So Labrador duck is this extinct bird, and there were eggs in various collections. And when he did the genetic work and was finished with it, he was able to show that none of these eggs were actually Labrador duck. In other words, he could actually show that they were other species. And so the interesting thing is we still don't know what a Labrador duck egg looks like based on the collections anywhere. But it was to me, a fascinating use of the ability to get usable DNA material out of egg collections.
That's a crazy story. Well, thank you, John. It was really very
My
interesting. And we'll have to have you back before 5 years and hopefully in 5 months. And I would encourage everyone who hasn't read The Feather Thief to read this, because you will just be sick about the collections that were lost in this.
Yeah, we could have a whole discussion about that. I mean,
Yeah.
we actually, I had some interactions with the author, who I have a lot of respect for. He's a very interesting guy.
Yeah.
And the first time I was introduced to him, he was visiting the collections of the Peabody Museum at Yale, and he sent me a picture of some eggs from our collection that they had just pulled off one of the dark web sites that the fish fly tyers use. And my heart just, couldn't believe it. But I looked at them, and there was 3 specimens of tanagers, and I was like, huh. And I looked at the tags, and they'd actually been collected by one of my graduate students only about 2 or 3 years before this message was sent. And so I went down to the collection, and I opened up the drawers, and they were sitting right there. And I think what goes on there is that sometimes people post pictures of our specimens on the web just because they think they're interesting, and people on these fish fly tying websites kind of latch onto them and say, look at this.
Wow.
But no, it's a very interesting story, and I mean, to me, it highlights a lot about what shouldn't happen to collections, which is that they shouldn't become dormant and put off in the countryside in the UK, which is what's happened with the Natural History Museum bird collection at Tring, which is a beautiful place. It's out of sight, out of mind. And one of the things that I think is most valuable about our collection is that we've got so much active research going on that there are people in the collections all the time, and that's important.
Well, we'll have to visit again soon. So, all right. Thank you again. Thanks everyone for attending. I want to tell you that everybody stayed, John. So that's a good.
Thanks, everybody. It's my pleasure.
Thank you. I'll be in touch, John.
Okay.
Bye.
Michael.
Thank you.
Hi, everybody. It was good to see everybody.
Great.
I'm here.
In
Thanks,
person
John.
next time, huh? Or
Bye.
soon, I hope.
Great presentation.